Hi students, I am G. Sai Chandrasekhar, lecturer in zoology, and in this chapter at a glance, now I want to explain biotechnological applications in medicine. And here actually biotechnological applications in botany as well as zoology, in both the subjects it is important, and by taking both into consideration they are giving around 3 questions, sometimes 4 also, but related to zoology they are giving on an average 1 question only. And if you take here, generally 1 question they are giving. NEET previous questions 2023 1 question, 2024 1 question, 2025 also 1 question. With respect to zoology we can expect around 1 question this year also. That means in total 3 or 4 questions in total topic of biotechnology and its applications, in which our topic with respect to medicine, here only they are giving 1 question. And here in 25 1 question is given, which of the following genetically engineered organisms was used by Eli Lilly to prepare human insulin, that is also called Humulin. And this human insulin developed by taking bacterium Escherichia coli. So that question, simple question is given in 2025, that means a single line question. Next one, if you take 24, match list 1 with list 2, here alpha 1 antitrypsin, cry-IAB, cry-IAC, enzyme replacement therapy, like the 2 points from zoology and 2 points from botany are given, and that is also very simple question. And this question is given, here it is the matching question. If you take 23 question, which one of the following techniques does not serve the purpose of early diagnosis of a disease for its early treatment? Serum and urine analysis, polymerase chain reaction, enzyme linked immunosorbent assay, recombinant DNA technology. These are the 4 points which are given. Actually serum and urine analysis is a traditional one, because of that it is the odd man out with respect to this question. And in this question PCR or enzyme linked immunosorbent assay ELISA, or if you take recombinant DNA technology, these 3 are commonly used in molecular diagnosis, while first one is traditional, because of that the first option. By taking all into consideration, we can expect very simple questions in the case of this chapter. But here the thing, there is a chance for asking some different model questions also beyond the limits, but they are not asking and they are confined to NCERT with respect to our area, which is given in rDNA technology, which is used in medicine. And here, to avoid mistakes, memory based questions can be forgotten easily. So we should focus more on them in the last 2 or 3 days. Since mistakes vary from person to person, we should revise the subtopics where we are more likely to make mistakes. Generally, if you take in this area, the subtopics, even though very simple, these are strictly memory based, because we have to learn clearly, and generally they are not giving assertion reason model questions in this area, statement 1 and 2 model questions also. So we can expect very simple questions from this chapter, and if any deviation is also present, you can do better, no problem at all with respect to this topic. Okay. Now if you take biotechnological applications in medicine, from here onwards it is related to zoology part, animal part. So by taking animal part, now I want to explain some important major areas which are little bit confused by you. And if you take, this is the area 30 recombinant therapeutics. Therapeutic means which is generally used for treatment. And by taking rDNA technology, the people developed around 30 components. Among them 12 of these are presently being marketed in our India like countries. Different varieties of components are present. For example, if you take erythropoietin or if you take the insulin which is given in our textbook, and also growth hormone, like varieties of components are prepared or manufactured by taking a bacterium, which is the miniature chemical industry. By taking bacteria into consideration, we develop different varieties of components. Among them, 30 recombinant therapeutics are available in the market in total world, while if you take India, accepted only 12. This is one of the important areas memorized because it is strictly memory related. Genetically engineered insulin. Insulin is a hypoglycemic hormone, and this hormone actually prepared by pancreas. Islets of Langerhans are present, in which alpha cells help in the production of glucagon and beta cells help in the production of insulin. This insulin is one of the important components that is essential to control, regulate the glucose levels in the blood. Uptake of glucose is increased. Uptake of amino acids is increased by this insulin, and such a type of insulin deficiency is present, and that may lead to age onset diabetes. Sometimes insulin deficiency occurs genetically, and that may cause the problem, namely juvenile diabetes. So, diabetes mellitus is the disorder that occurs due to insulin deficiency. And here some of the important points: what would a diabetic patient do if enough human insulin was not available? Here the two different things previously used one. For example, if you take the insulin from other animals, because almost all the vertebrates evolved from the same common ancestor, because of that the production of insulin is very common, and the insulin of any other organism can be used by man. But the extracted insulin, if used by man, there is a chance of allergic reactions for some people. From slaughtered houses, especially from pigs, pancreatic islets are extracted, or if you take bovine insulin extracted from cow and calf, and in such a type of areas, if the components are collected, there is a chance for allergic reactions. So, immune responses may present, and they may cause some other problems to the people. That is the major. So, that question is given: would the insulin isolated from other animals be just as effective as that secreted by the human body itself? Would it not elicit an immune response in the human body? It exhibits immune response 100%, no doubt. Because of that, the bacteria, from the bacteria we are preparing the human insulin directly, and that is used by us. And if you take here, think about whether insulin can be orally administered to diabetic people or not. Why? That question also present in NCERT book. Insulin is a protein hormone. For example, once we take insulin into our mouth, if it is taken into our alimentary canal, in the stomach as well as in intestine, you can observe the proteases. They help in the digestion of the proteins. So, denaturing tendency is present for the insulin. What the insulin taken by us cannot be taken into the blood. Because of that, there is a problem. So, we shouldn't take insulin orally. So, we have to administer into the body, intramuscular or intravenous style. Next one, insulin used for diabetic was earlier extracted from pancreas of slaughtered cattle and pig. Bovine insulin, porcine insulin. Bovine insulin means from cow and calf like animals, and if you take pigs means porcine, P-O-R-C-I-N-E, porcine. And here some patients to develop allergy or other types of reactions to the foreign protein. That point is given. And actually insulin is just like an enzyme. Pro-enzyme is present, inactive form of enzyme is present. Like that, in the case of insulin also we can see the pro-insulin. The pro-insulin has 3 peptide chains. A chain is present, B chain is present. These two are very, very important for the formation of a mature insulin, and these two are connected by C-peptide. So, C-peptide is present between A peptide and B peptide, and that is called immature insulin, which is called pro-insulin. And in this pro-insulin, we can observe C-peptide is to be removed for the maturation of insulin. And in our body, one gene helps in the production of A, another one participates in the formation of B. At the time of synthesis, it will become difficult from the bacteria. In rDNA technology, even though the production of the different varieties of components taking place with the help of bacteria, sometimes it will become difficult. And in the case of insulin, because A peptide is present, B peptide is present, so these two are to be synthesized from two different bacteria separately, and they are to be connected later with the help of the bonds. They are called disulfide bonds. So for this purpose, here the synthesis will become little bit difficult. So some points, highlighted points are present here. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by disulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone, that I told now, and if you take, contains an extra stretch called C-peptide. The C-peptide is not present in the mature insulin and is removed during maturation into insulin. So this point is very, very important. The main challenge for production of insulin using rDNA techniques was getting insulin assembled into a mature form. Which one is the difficult task for the people to produce the insulin in the lab? So that question may be asked in exam. So at that moment you have to write. In 1983, Eli Lilly, 2025 question. Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of Escherichia coli to produce the mature insulin directly. And they also created disulfide bonds and they connected them. And if you take here, in the case of this one, the disulfide bonds are developed between cysteine and cysteine of A chain, one that is called intradisulfide bond. Intradisulfide bonds are present between 7 and 7 of A and B chains. Next one, namely 20 and 19 cysteine of the A and B. So 3 disulfide bonds are present. So how many disulfide bonds are present? The question is present. In between which amino acids you can observe the connection is present. It is little bit out of the box question. And if you take pro-insulin, A peptide, B peptide, how the connection, this is present in textbook also, you can understand it carefully. That is about first one, namely insulin. Next chapter which is given here is gene therapy. And this gene therapy, just a treatment by using the gene. Previously, any genetic disorder is present to a person, there was no medicine at all and there was no treatment also. But today we can observe this gene therapy. And in the case of gene therapy, if a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. That point is given in our NCERT. So gene therapy can be done at the time of childhood or embryonic condition also. For example, if you take here, there are two different methods are present. Somatic cell gene therapy is present, where the treatment is done with the somatic cells. In the somatic cells only the treatment is done. And germline gene therapy is present. That means either in sperm or ovum, in other words gametes, and also in early embryo, if you do, it will become comfortable and also successful. So even though more success is present, because of ethical, legal, social issues are related to it, germline gene therapy is in infant stage itself is present now. And if you take somatic cell gene therapy is commonly conducting gene therapy, and now we are thinking about one of the important gene therapies which was conducted. The first clinical gene therapy was given in 1990 to a 4-year-old girl, Ashanti De Silva. That girl named Ashanti De Silva. 1990 to a 4-year-old girl with adenosine deaminase deficiency, ADA deficiency. What is the problem with respect to ADA deficiency? It is not present here, but we have to understand it. ADA deficiency leads to severe combined immunodeficiency, SCID. It is a kind of primary immunodeficiency. And in the case of SCID, even though in bone marrow the B cells and T cells are formed, they are unable to mature. For the sake of maturation, ADA enzyme is required, and these people do not have such a type of enzyme. Because of that, the problem arises. So for the sake of that one, by hook or crook, we have to introduce ADA enzyme into them for the maturation of the lymphocytes. There were different treatments were present. First one, namely bone marrow transplantation, and 2024 I think enzyme replacement therapy is given, and these treatments are generally used for the treatment of ADA enzyme problems. But when compared with those, somatic cell gene therapy is present, and that is one of the best methods which is discovered, even though it is not 100% perfect. But if you take at the time of embryonic condition or at the time of gametic condition, if you do, it is successful. But because of ethical, legal, social issues are there, we are thinking about somatic cell gene therapy, also ex vivo. Ex vivo means outside the body. And here first we are extracting the lymphocytes from that particular child, where already we can observe the deleterious gene related to ADA. ADA gene is not functioning in her or in that child, and that is collected. And whenever this is collected, these lymphocytes are collected, and to which we are introducing the gene related to ADA, and they will become therapeutic cells. We are extracting from the bone marrow the lymphocytes, and into them we are adding this component, namely gene. In different methods are present, you know, in botany they explain different methods are present, where using plasmids or using cosmids, or if you take BAC or YAC, like different varieties of technologies are present, or introduction micro injection is present, like the gene gun technique is present, varieties of things are present. By using different methods, we are introducing genes into them. Then what happens? Normally they will become healthy. These lymphocytes are pumped into the child and they will become normal. But here the problem is the lymphocytes are not immortal. They exhibit death. Lymphocytes have their own lifespan, and after the completion of their lifespan they are destroyed in our body. Because of that, again and again we have to transfuse them. That is the problem, and this is known as bubble baby syndrome also, or simply bubble boy syndrome also. And this disorder, they have to be kept in special area, where in a protecting zone, for example if the lymphocytes are absent. So we are using lymphocytes and we are protecting the children from such a type of disorders. So gene therapy is one of the important areas. Here the main points which are present here: first clinical gene therapy 1990, the year memory based, 4-year-old child, that is memory based. Adenosine deaminase deficiency, that is the first clinical gene therapy conducted, that is also important. And what are the alternative methods present for this ADA? Bone marrow transplantation and enzyme replacement therapy, and in which we are introducing the gene, therapeutic gene, lymphocytes, which are present in bone marrow. Such a type of questions are very, very important, so we have to think about them carefully. And here ADA is introduced into the cells at early embryonic stages. It could be a permanent cure, and that is also very, very important. We have to memorize this area. Next, molecular diagnosis is one more important subchapter. And if you take in molecular diagnosis, some of the words are present, we have to memorize carefully them. For example, pathophysiology. What is pathophysiology? Most of the students are confused here. Pathos means disease. For example, any microbe enters into our body or any ailment occurs to our body, the physiological changes occur in us. That is, they are studied under pathophysiology. For example, a normal person, the person who is suffering from typhoid, if you take the typhoid person, he is different with respect to his metabolism. He is different with respect to his activities, and that is studied in pathophysiology. So pathophysiology is the word very, very important here. Using conventional methods, previously we used to, generally in our country like countries, we can see normally we are going with the traditional. For example, if you take this one, namely for typhoid, also we are conducting the tests, Widal test is present. Or if you take normal blood analysis, for example jaundice is present, urine analysis or serum analysis. These are all common traditional methods, but we are unable to discover in early stages. For the discovery of the disorder in early stage, molecular diagnosis is very important. What are the different techniques present here? rDNA technology is one important one, polymerase chain reaction PCR is second one, and enzyme linked immunosorbent assay ELISA is the third one. These are the three important varieties of methods which are present to identify the disorder early stage itself. And if you take here, presence of a pathogen is normally suspected only when the pathogen has produced a disease symptom. This is common traditional method. But if you take in this one molecular diagnosis, early detection is possible. And if you take here, especially in the case of AIDS patients, we are not only using ELISA, that is the screening test, but also we are using PCR, polymerase chain reaction, which is also called people's choice reaction, and this PCR is now routinely used to detect HIV in suspected AIDS patients. That means not only ELISA, which is used as a screening test, this is the routine test which is used for identification, RT-PCR test. And also, if you take one more important but last point which is given, it is being used to detect mutations in gene suspected cancer patients too. It is a powerful technique to identify many other genetic disorders also. Such a type of words are present in these simple sentences. Even though the chapter appears, subchapter appears a few lines, each and every line is valuable, so read carefully and get clarity related to the lines. Next one, if you take here a single stranded DNA or RNA tagged to radioactive molecule. For example, if you take a radioactive molecule is added to the single stranded DNA, to which we are using a probe, that is the complementary unit for this DNA strand, and when these two are associated with each other, if any mutation is present related to genetic problem or related to cancer problem, there the complementarity is not possible. Because of that, it can be identified with the help of autoradiography. By taking X-rays, we can identify the area and that can be detected where the problem is present. That point also given in a simple line. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. That point is given. So, very simple single line is given, but here by understanding rDNA technology you can notice it. That means by taking single stranded DNA or RNA on one side, it is also with radioactive element molecule is used and a probe is used which is complementary to this one. Complementarity is possible in all the places if it is healthy. If any problem is present, there the mutation is present, there is no chance for the connection. That means complementarity is not possible and that is detected. Technique is very simple, doing is difficult, that is all. So, like that, this is the important point, and ELISA is based on antigen antibody interaction. That means we can detect the antigen which is present in a person, that is called direct ELISA. For example, if we detect the antibody in the person, that is called indirect ELISA. So, ELISA, two different techniques are present: identification of antigen by using antibody or identification of antibody by using antigen. The two different methods are present, they are called direct ELISA and indirect ELISA also. And infection by pathogen can be detected by the presence of antigens or by detecting the antibodies. Antigen detection is called direct ELISA, antibody detection is called indirect ELISA. These 2 points are not given in NCERT, it is little bit beyond, but they are important in examination point of view. Next one, transgenic animals. The definition is very important in transgenic animal because one animal has one special DNA component in its body. If we add the DNA of some other organism, a foreign DNA, then it is called transgenic animal. Animals that have had their DNA manipulated to possess and express an extra or foreign gene are known as transgenic animals. That word is given. 95% of the transgenic animals developed are nothing but mice because their lifespan is very short. That means the mouse can that means if it is pregnant, the gestation period just 28 days. So, maturation also earlier. That means number of generations we can identify, number of organisms we can develop. Because of that, mice are taken as the classical examples, and they are amicable or adjustable to develop the transgenic animal. Next one, what are the important points in the transgenic animals? Why we are using these transgenic animals? First one here, related to normal physiology and development. Normal physiology and development. For example, if you take transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. For example, human growth hormone related components are introduced into a particular, that gene is introduced into a mouse. We can get super mouse. Brinter and Palmiter like people developed it. And if you take here, we can observe growth, insulin like growth factors related to human, if introduced into that mouse. What is the difference between normal mouse and the super mouse? We can identify. And physiological aspects, how are taking place, and what are the changes occurring can be identified. This is the first important point we have to understand related to the benefit we have to understand. Second benefit is normal physiology and development, study of diseases also. For example, if you take different varieties of diseases can be identified, especially cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's like. Cancer, it is a cell-based disorder. Cystic fibrosis, it is a genetic disorder of 7th chromosome. Or rheumatoid arthritis, it is an autoimmune disorder. Alzheimer's, age onset memory loss, all these things are present in Alzheimer's. In all these cases, the disease symptoms how occurring and how can we treat them, what are the important changes occur if the disorders occur, and all these things can be studied. That is the second one. Next one, namely, if you take third one, biological products. For example, if you take biological products also developed. If you take alpha-1 antitrypsin, that is one of the important proteins present in our body that maintains the alveoli of lungs. For example, if you take a person who is continuously smoking, or if any dust particles enter, then we can observe the lung problems arise. In those cases, we can observe the problem arises called emphysema. This emphysema can be treated with the help of alpha-1 antitrypsin that is developed in a particular sheep by name Trypsi, Trypsi, Trypsi. In that animal, we developed that one. And also if you take for phenylketonuria, cystic fibrosis like genetic disorders, what are the products which are essential to control the disorders are identified. And one of the greatest changes which developed in the case of this one in 1997, the first transgenic cow Rosie produced human protein enriched milk 2.54 grams per liter. These points are very, very important in examination point of view. The milk contains the human alpha-lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Breast milk is the best milk. Mother milk is very, very important for the child. If mother is unable to give the milk to the child, we are taking into consideration either buffalo or cow. What the components required for the growth of the child are not present in that buffalo milk or cow milk. By the introduction of this alpha-lactalbumin, development of Rosie like cows, they are equal to our mother's milk. Because of that, now this cow milk, Rosie milk is equal to the mother's milk. So the child growth as like the normal other conditions here we can see. So for the sake of that one, it is very, very important. Next one, namely, for example, if you take vaccine safety, generally to detect the problems related to the introduction of a vaccine, we normally use some other animals as scapegoats or the animals which are used, that means guinea pigs. And if you take here, generally monkeys are used as scapegoats for them previously, and their number also might be decreased. if you do like this. So we developed that means transgenic animals, especially transgenic mice. If they replace the monkeys, 100% success will present with respect to vaccine safety and other things also. We can protect the other animals also, biodiversity also. So for the sake of that one here, and transgenic mice are being used to test the safety of the polio vaccine. Like that, not only for polio vaccine, different varieties of vaccines can be observed what the evil effects occur by the introduction of that vaccine into our body. Instead of using the monkeys, if we start to use mice, it will become protection for the monkeys also. So vaccine safety, for that purpose also transgenic animals can be used. Chemical safety testing also. Here one line is given, transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain results in less time. Next, ethical issues are also present. Here in the ethical issues, especially here that point, Indian government has set up organizations such as GEAC, that point is very, very important. The modification usage of living organisms for public services as food and medicine sources, for example, has also created problems with the patents granted for them, for the same. So like some points are given, and here we have to think about these ethical issues also. With that we completed this chapter. Here, not to be missed, which points are not to be missed here? For example, why some of the people get allergy by taking insulin from other animals? That point may be asked in examinations. So allergic reactions are developed, because of that we cannot take directly. That is the major problem. Insulin cannot be taken orally because it is digestible, because it is a protein. Next one, why A and B chains are, of peptides are developed separately? Because in our body also two different genes participate in the production. So we have to develop the two different genes separately in two different bacteria, and again they are to be connected with the help of disulfide bonds. In gene therapy, the transfusion of therapeutic lymphocytes repeatedly. Why? Because they are not immortal. They are mortal, they die, they have one span, lifespan, after the lifespan they die. So that is also one more important point you have to understand. Which method of molecular diagnosis is powerful to identify genetic disorders? PCR is given, polymerase chain reaction, so that line also very important. What is the principle of ELISA? Antigen antibody reactions. Detection of antigen by using antibody is called direct ELISA. Detection of antibody by using antigen is called indirect ELISA. So two different methods are present. That is the principle. Why the milk of Rosie is better than the other cows means it is almost equal to mother's milk. Just like breast milk, it is also almost similar. Next, what was the benefit we can get if the mice replace the monkeys? We can protect the monkeys. So like some important areas are present here. Even though the chapter appears to be little bit confusing, if you read carefully you can get good marks. And I have given some questions also, around 20 questions are given. So carefully go through these questions, you can understand the topic in a clear cut manner. All the best.